and it's my pleasure and honor to introduce Courtney Fitzpatrick, Dr. Courtney Fitzpatrick. She has an academic history that kind of foreshadows this very symposium. She started off her undergrad at UNC Chapel Hill in biology and then quickly turned a left turn into art, and she actually graduated with a degree in art. She then did some social work and then got the hankering to go back into biology. So she started to take more classes and did some research assistant work. Then she got her PhD at Duke and did some field work in Kenya. She's done work on a lot of amazing systems, including baboons, lemurs, elephants, and vitamin B. She's also done field work as well as a lot of theoretical work. And now she's working as a postdoc with Mike Wade at Indiana University, and she has some lizard work coming up in the future, so stay tuned.
Great, thank you. Can everybody hear me okay? All right, thank you for that nice introduction, and I'm excited to be part of this symposium. I've had a blast already, and it's only 11 o'clock. I've enjoyed talking to everybody and look forward to more. So, the ecology and evolution of color, for a lot of us, for those of us who focus on the biology of animals, I think often what the phenotypes that pop to mind when we hear about the ecology and evolution of color are these flamboyant phenotypes that we see in males of so many species. And so these have been and continue to be really well studied, and they're typically understood, not always, but typically understood to be the outcome of sexual selection. And this is a process that everyone is familiar with, but just to be really explicit about what I mean when I say sexual selection, I'm going to review this process. So this is when these conditions are met, right? If there's variance in the mating success, so some individuals get a lot of mates, few or most individuals get few or even no mates. And if that variance in mating success predicts variance in relative fitness in the population, and if having some exaggerated colorful phenotype predicts where an individual is going to fall in the distribution of mating success, then we should expect to see the evolution of colorful sexual signals. And that's what we see so often. And I'm just throwing up some of the classic examples here, and each of you can probably come up with 10 more off the tops of your heads. But what's becoming increasingly clear in recent decades, and what I'm going to focus on, is that females across taxa have also evolved colorful ornaments. And it's important to say at the outset that these are in species that are not just sexual reverse species. So I'm going to tell you my agenda. So my agenda for this talk is my message has 3 parts. The first part is that colorful female ornaments are not that rare. The more we look for them, the more we find them. And if we don't understand their ecology and evolution, then there's a non-trivial piece of biodiversity that we don't understand. And then I'm going to use some of the theoretical work that I've been doing to try to convince you that it's looking like my work is consistent with some work that's been done by other people that suggests that the processes that give rise to these ornaments in females differ in some fundamental ways from the processes that we understand pretty well that give rise to the ornaments that we see in males that have actually already been discussed nicely this morning. And then if that's the case, then I submit that they are a good model for understanding social evolution more generally. And when I say social evolution, what I mean is understanding how the fact of being social constrains or facilitates trait evolution. So if I haven't convinced you already that female ornaments are not rare, let me just do a little tour of some organisms here. So my original empirical study system, baboons, have these exaggerated estrus swellings. I'm going to talk a little bit more about those. Striped plateau lizards have orange patches underneath their throats during the breeding season. The belted kingfisher, when you see a belted kingfisher with a rusted belt, that's the female. Blue crabs, the females get red pinchers during the breeding season. The males do not. And it's been shown that males prefer females with redder claws. Two-spotted gobies I'm going to talk about a little bit more. So I'm going to, whoops. And empid flies. Empid flies, long-tailed dance flies, have these orange abdominal air sacs that they inflate and display during the mating system. These are all mating season. These are all females, in case that's not obvious. And when we start to look at these traits as a sort of on the whole, two general patterns, I should call them patterns rather than rules, start to emerge. So what I'm showing you here are examples of ornamented females paired with a male, not necessarily from the same species, but the same general taxon. So, so one of the patterns that starts to emerge is that they tend to be less flamboyant than what we see in males. So I'm not making the claim that you see anything like a peacock's train in females. Just to make this a little bit more clear, so these exaggerated estrus swellings, they're conspicuous, definitely visual signals, but they pale in comparison to what we see in male mandrills. The two-spotted goby females get these orange lateral spots during the breeding season. It's been demonstrated that males prefer females with bigger orange spots, but they're nothing like the guppies that we've heard about this morning or like Molly's swordtails. I'd love to borrow one of your pictures for this slide. This is just a Wikimedia guppy. And then the striped plateau lizard female has this conspicuous signal, but it doesn't reach the level of intensity that we see with many other phrynosomatid lizards. And the second thing is that they tend to be dynamic, so they come and go through the life of the organism. It's not the case that this is never true for males, but with the exception of the kingfisher, it's always true for what I've been able to find for females, where they generally show up around the time of the onset of receptivity or the breeding season, mating season, depending on the biology of the organism and the mating system. Nonetheless, so they don't reach the level of intensity that we see in males. They come and go. Perhaps this is why they've kind of flown under the radar a little bit until recently. Nonetheless, they should be costly, right? They should expose individuals to increased levels of predation, increased risks of predation. They should impose metabolic and energetic costs. So they require some kind of evolutionary explanation. And two explanations might pop immediately to mind. One I've already touched on, which is perhaps these are sex role reverse species. Maybe the females have that variance in mating success, and maybe that variance in mating success predicts something about relative fitness, which we know is the case for sex role reverse species. All of the examples that I've shown you so far are in species that we think of as having traditional mating systems. This is an unfortunate phrase that we use, but everybody in this room knows what I mean by traditional. So that's not a sufficient explanation. But maybe there's a genetic correlation. Maybe these are traits in females that are genetically correlated with traits in males that are under really strong sexual selection. That's a thing that we know happens in the natural world, but all of the examples that I'm talking about are cases where it's female-biased expression. So there's not a similar trait in the males. So that's an insufficient explanation for the full range of variation within and between species. So a third explanation that has been kind of out there in the literature is that maybe females are competing, if they're not competing for number of mates, like we know is so often the case for males, maybe they're competing for males of superior quality. You can define quality in a lot of different ways. Sometimes we are explicit about it and sometimes we are not. But that is the question that got me started with my dissertation work. I'm just going to touch on this empirical work that inspired the mathematical modeling that I've been doing more recently. So I started out trying to understand these exaggerated estrus swellings in baboons. These are edemas. They show up around a little before the time of ovulation. They tend to be biggest around the time of ovulation, and then they disappear quickly, and they keep coming and going until the female conceives. I was doing this in collaboration with the Amboseli Baboon Research Project. So this is a wild population, long-term study. And these are, so for the color symposium, I want to make sure and say, these are definitely visual signals. That's not to say that there's not some olfactory component, but they did crazy experiments in the 60s where they overectomized females and strapped on prosthetic swellings, and the males responded to them. Males in captivity respond to photographs of females with swelling. So they're undeniably visual. And often they are, I have this here to remind myself to tell you they're often used as the textbook, literally the textbook example of sexual selection in females. And in this case, the sexual selection hypothesis tends to be that females are competing for males of superior quality. So if there are any primate people in the audience, you might recognize some of these hypotheses that I have listed here. These are the explicit hypotheses, the best male hypothesis, the reliable indicator hypothesis, the graded signal hypothesis, indirect female choice hypothesis. These all differ in some ways, but at their core, they all hypothesize that females are competing for males of superior quality. So that's one of the things that I set out to test. And so the prediction, right, is that females with more prominent swellings should be mated by the highest ranking male, which is the male, the highest quality male in a primate social system. And I'm gonna just gloss over a lot of the methodological details and just tell you that we found no evidence for that in this study. Because of the power of the study system, we were able to do the most complete test of this hypothesis. So on the x-axis is swelling size in millimeters. On the y-axis is the probability that a female is mated by the highest ranking male. And the dashed line and solid line and open and closed circles are just two different measurements of size, right? So there is no evidence that females with more prominent swellings are more likely to mate with a dominant male. So at least in this one study system, in this one population at this one time, we found no empirical evidence in support of this idea that females are competing for higher quality males. And this got me thinking about the question in principle. So how much potential is there for variation in male quality to drive the evolution of female choice? And that, in principle question, is a theoretical question. So I'm gonna transition to talking about the theory part now, and I just want to make the point that the purpose of this kind of theory, this is my main collaborator on this is Maria Servideo. She calls these proof of concept models. And this is from a paper that we wrote, that theory's role is to make sure that hypotheses are logically consistent in contrast to empirical research, which was required to actually tell us about what's going on in the natural world. So another way of saying that is that proof of concept models test the validity of a verbal model. Okay, so this is the question that I started out with. Does the combination of choosiness in males and variation in male quality, is that sufficient to drive the evolution of female traits? And I'll give you the answer before I tell you how I got to it, which is that we found our model suggests that it's not. So I'm going to step through the model, and this is just a basic population genetics model. If you're familiar with Kirkpatrick 1982, this is very much in that vein. I am not a theoretician's theoretician. I'm an organismal biologist who uses theory. So this is not a very complicated model. So it's haploid, it's polygyny. So all females mate and all females have equal mating success. And we did that specifically because we don't want to ask about the effect of the variance in mating success because we already know that. We want to ask about the potential for male quality. And there are three loci. There's a trait locus that's expressed in females. There's a preference locus that's expressed in males. Choosy males prefer ornamented females. And then there's this B locus, which is quality. And in this case, we're modeling quality as a direct benefit, which should be actually stronger than a good genes model. So this is, in this case, females who mate with high quality males have higher fecundity. They have higher reproductive success. So I'm just using these cartoon crabs to make the point that with 3 loci, 2 states at each locus, there are 8 possible genotypes. In this model, that translates to 2 possible phenotypes for females. Females can be ornamented or unornamented. And 4 phenotypes for males. Males can be regular males. They can be high quality males. They can be choosy males, or they can be choosy and high quality. We stop at 11:25. Okay. So this is the life cycle. One turn of the life cycle is represented here. In the first step, there's natural selection on the female trait. So the females experience a cost as a consequence of having the trait. The next step is the mating step. And then the final step is birth, recombination, and mutation. And the mutation is just in there for logistical purposes. So if you'll indulge my sort of childish metaphor for a minute, this is my Dr. Seuss representation of how these kinds of models work. It was not obvious to me when I started out how they work. So this is what you do. This is the model. You build the model, and then allele frequencies go in, and then allele frequencies come out. So the input are allele frequencies, the output are allele frequencies. You can change the knobs to represent different values of your parameters and turn the crank for as many generations as you want. And now I'm going to step through the life cycle again to tell you a little bit about each of the important parameters. So the first one is very simple. It's just some selection coefficient. There's selection, natural selection on the female trait. The mating step, there are a number of things that happen. Choosy males prefer ornamented females by some amount, AM. High-quality males can compete more intensively by some amount, 1 plus gamma AB. But just think the AB is the magnitude of the male quality. So higher quality males can compete more intensively, and then that'll show up again in the birth step. And then there's this interaction term that becomes important at the end, rho, which is an interaction term that we can turn on or off, mediating the relationship between the P and the B locus. And then finally, during the birth, the fecundity step, ornamented females have increased fecundity on their own, and I'm going to ask you in the interest of time to just take it on faith that, and if anybody's interested in the details of this, find me afterwards and we can talk about it, but this is required in order to get the model to run. Maria Cervedio and Russ Landy have shown, and it's consistent with all the empirical research, that male mate choice in polygynous populations can't be maintained unless it's as a function of some trait that's related to female fecundity or readiness to mate. So I have this in gray just to show you that it's here, and because I'm going to need to refer to it later, but it's not actually the parameter that we're interested in. What we are interested in is this quality locus in males that confers a fecundity benefit on the pair. So a female mated with a high quality male has more offspring. So the first thing that we had to do, we explored the parameter space and found some value at which the T2 allele, the allele for the female ornament, is polymorphic, so that it's not at 0 and it's not at 1, because we wanted to ask what happens if we turn the volume up and down on these parameters. We didn't want to be at a ceiling or a floor. So this horizontal line is just that point of comparison. And then we can ask, when this is that point of comparison when there's no quality and there's no male preference. And then we can ask, what happens as we turn up the strength of the male preference? So this is the strength of the male preference on the x-axis, and on the y-axis is just the equilibrium value after 1,000 generations. I should say we analyzed this with simulations because the normalizations were too complicated to get an analytic solution. So these are all simulations. We let it run for 1,000 generations and it reaches equilibrium. And there's essentially no difference. And in fact, the negligible difference that you see when the strength of the male preference is turned up is an artifact of the unidirectional mutation that we had in the model as a logistical parameter. So there's no effect of male preference. Similarly, there is no effect on the magnitude. The magnitude of the male quality has no effect on the final equilibrium value for T2. So these two parameters are they're not driving anything about the evolutionary trajectory of the female trait. And this, it's one of those hindsight things. It makes sense in hindsight because there's nothing biasing the high quality males in this form of the model. There's nothing biasing the high quality males towards ornamented females. So then we said, well, what if we turn on this interaction term so that in the way that this interaction term functions effectively is to make high quality males even more successful competitors than they would be if they weren't choosy. If they weren't choosy as well, right? So a male who's both high quality and choosy sort of technical, but it has like above multiplicative mating success. It is a technical, it is a statistical interaction term. So if that's the language that makes more sense to you, you can think of it that way. So, and what we found, I just want to orient you here. This is each one of these diptychs is the same diptych, is similar to the diptych that I just showed you. So on the left is the strength of the male preference. On the right, each one is looking at the magnitude of male quality. Each panel here is just a different slice of parameter space. So it's just a different balance between the selection on the female trait and the fecundity benefit associated with the female trait. And the message, and the y-axis is the difference between the equilibrium value when the interaction term is turned off, and then what happens when you turn that interaction term on. And we thought that what we were going to do was show that, well, I'm not going to say that. I'm just going to say what we found was that it's actually a very weak effect. Even when we rig the system so that high quality males are much more likely to mate with ornamented females, the effect is weak. And in fact, in most of the parameter space, it's negligible. So this was the strongest effect we could find. It's only a difference of 0.1. Now these are descriptive, right? It doesn't make sense from a model like this to draw really strong quantitative conclusions, but it's just not a very strong effect. And the difference in the closed circles and the open circles is just two different values for that rho term. So we looked at rho as 1.5 and rho is 2. Okay, great. So that doesn't bode well for the third potential explanation for what's driving the evolution of these traits in females. So the conclusion from that model is that unless some feature of the biology links male fecundity benefit to male choosiness, which, by the way, we don't know how common that is in the natural world, we should not expect male quality to shape female traits. And even when there is some kind of link, like that rho term, the effect is pretty weak. So what is the explanation? What gives? And what I have turned to recently is this idea that is not my own, but I am very sympathetic to, is an idea put forward by Mary Jane West Eberhard in the late 70s and early 80s about social selection. That social selection, which is competition for resources that are mediated by other individuals, by conspecifics, is likely to be a very important force for females, potentially more important for females than for males. This diagram is adapted from a figure that was in a special issue of Philosophic Phil-Trans. And what the idea here is that social selection is a subset of natural selection, and that sexual selection is a subset of social selection. And I'm saying that for people who might be familiar with this literature. I want to make sure and recognize that I'm not making some claims about them being, totally different things, because that can become and has become a semantic argument that's not productive. But the idea is that females are more likely to be competing using a sexual signal, what looks like a sexual signal, to obtain social benefits like protection from predation, access to resources, or parental care, even if the parental care is only avoidance of infanticide. I always say that, like, not killing the kid is it's like the lowest threshold for paternal care. So I already said this. So the idea is that females are using a sexual signal for social benefits. And there's a lot out there that we don't know. I mean, this is just, Talia so nicely remembered that I've got this lizard project in mind that I want to start working on. There's tons of variation, and a lot of it is undescribed, actually. And phrynosomatid lizards is just one group of lizards. Many species, many closely related species. There's variation between species, lots of variation within species. And thank you. That was kind of an abrupt end, but I'm terrified of going over.
So in some butterflies, some color pattern polymorphisms are thought to evolve in females basically females maybe mimicking males as a way of avoiding harassment by males.
Yeah.
Like, and I just wondered to what extent the fact that the signals you're looking at are really, they're present when the female's receptive,
Right.
when she wants to mate, but then they go away. I wonder if there's any connection there with perhaps them being a signal of interest, and then they're sort of like an honest signal and maybe a way of avoiding
Absolutely.
male interest, male harassment outside of breeding time.
Right. I mean, the thing is that we know that we know that female organisms can signal receptivity. Like, they don't need to exaggerate it to signal receptivity, because plenty of organisms do that. So the question is, why exaggerate it? And in the case of the butterflies, and that's also, I mean, it's exciting to me because even the hypotheses are still really being formulated. But in the lizards, some lizards have breeding color that is when they're gravid. And so people have hypothesized that that's a harassment avoidance strategy. The baboons have a pregnancy signal. We have no idea if it's adaptive or not. In the butterflies, so they're mimicking males, right? So, I mean,
That's what they say.
the color patterns are similar to the ones that they look like males, yeah. So that is sort of easier to explain than these cases where it's a female-specific trait. But yeah, the harassment avoidance is surely really important.
Maybe you can say a little more about the hypothesis you presented at the end. My question is, if your hypothesis about social selection is an explanatory paradigm, then wouldn't you expect some of these visual signals to be present in circumstances other than receptivity? Because that is all tied to mating.
Yes.
But these resource acquisition
yes.
scenarios are present throughout much of life.
Yes,
So.
Thank you for saying that, because it reminds me of something I forgot to say, which is something I'm very passionate about at the moment. So I was so worried about going over that I forgot one of my main points. So if it's true that these mostly evolved from social selection, then I would submit that they are a good model, that it's a signature of a process that is undeniably important in all kinds of other parts of the life history. And that we don't know very much, like unlike sexual selection, where we have some ideas about when we should see really strong sexual selection under what demographic and ecological conditions, we don't really have an idea of when social selection should be really strong and when it should be less strong, even if you control for sociality, degree of sociality. So yes, I don't mean to be saying that that you shouldn't see any kind of signaling or consequence of social selection outside of the breeding context. But it is the case, I mean, I'll just speculate. In polygynous systems, males are very easy to manipulate. Males are under strong selection to maximize their mating success. So it might be the case that in highly polygynous, in systems where males are really constrained, females have a much greater opportunity. And so that might facilitate trait evolution. Yeah. Do we have time for one more? Or?
I think we have time for one more question.
So a lot of male sexual signals, people talk about how maybe the male signal evolved in the context of male-male competition, but now females pay attention to it too. Do you think a similar thing could be going on with female signals?
Yep. Yeah.
So,
it's just, there's so many empirical questions. So specifically especially in the lizards, where they are some territorial species, you can imagine that females might be using signals to compete with each other and it's certainly true, like I'm thinking of Kim Roswell's tree swallows. I mean, it's not a visual signal, but that aggression has evolved as a consequence of strong selection on females to compete with each other for resources. So if there are ways that females can use these visual signals to compete with each other, then that's absolutely part of the story. Yeah. Great. Thank you.